Hello students, welcome to EPG Pathshala. I am Dr. P.M. Reddy from the Energy and Resources Institute. Today we are going to discuss about a module on metabolic engineering and industrial products under the paper Plant Biotechnology and Crop Improvement. The learning objectives of this discussion are to familiarize with the metabolic engineering approaches adopted for amplification and or new production of valuable metabolites. Secondly, to review applications of metabolic engineering in the production of plant secondary metabolites, edible vaccines, industrial enzymes, and therapeutic proteins, and modification of existing pathways to enhance synthesis of high-value chemicals, as well as ways to engineer biofuel feeds to boost and the efficiencies of biofuel production, etc. Thirdly, to comprehend innovative metabolic engineering strategies for incorporating new biological pathways to improve plant performance. Metabolic engineering is defined as the directed improvement or manipulation of cellular processes through modification of specific biochemical reactions or the introduction of new ones for production of valuable metabolites. Most commonly, bacteria, yeast, or plants are used as a host organisms for the production. Metabolic engineering uses algorithms to mathematically model metabolic networks, automated design and evaluation of pathways to calculate yield of useful products, and also to discover network that restrains the production of the products. Metabolic engineering approaches are used for or involve amplification of enzyme levels, use enzymes with the different properties, addition of new enzymatic pathways, and deletion of existing enzymatic pathways, and so on. What are the requirements for the overproduction of desired product? First one is we should have a knowledge of pathways and the alternative to these pathways that can be used for desired production formation. The reactions should be thermodynamically feasible under biological conditions. And finally, the intermediate metabolites must not be toxic, and the host organisms should express the required enzymes for the conversion to take place. Computational and evaluation of existing and novel pathways. In 1990s, the first computer-based systematic construction of alternative non-obvious pathways for lysine production was published. The algorithm could identify and bypass the bottlenecks of the pathway and the underlying limitations that govern the biochemical pathways. Subsequently, in 2000s, a generalized algorithm, BNICE, that is Biochemical Network Integrated Computational Explorer, was developed to design new pathways based on structure of substrates and products. BNICE was used recently for the automated design and evaluation of novel biosynthetic routes for the production of hydroxypropanoate for butanediol. This is one of the very important compounds used in chemical industries. There are several applications on metabolic engineering, and some are given in this figure. Some of the most important ones are production of plant secondary metabolites, such as alkaloids, flavonoids, etc. production of edible vaccines, industrial enzymes, therapeutic proteins, antibodies, and recently scientists started using more complicated metabolic engineering for increasing photosynthetic efficiency, nitrogen fixation, nutritional quality, and biofuel production in plants. These applications of plant metabolic engineering nowadays, besides what I mentioned earlier, also includes development of resistant crops as well. New innovations have been incorporated into metabolic engineering to improve the efficiency of the production of metabolites. Some of these include development of fusion proteins, which reduce diffusion limitations. A product which is being formed should not be diffused out, and improves protein-protein interactions so that purification process becomes much easier. That's one of the benefits. And also, for increasing the pathway control, scaffolds with improved control over architecture has been developed, whereby all enzymatic reactions can occur in a confined manner, where the substrates and enzyme molecules are targeted in the close vicinity. And also it has a control over enzyme stoichiometry. And the third strategy which is used in new strategies, subcellular localization in organelles. This is capable of selective transport into compartment. Both substrates are being transported inside and the product is transported out. And intermediates are being retained inside the organelle so that they can be efficiently be used for the production of products. And also the intermediates are prevented from escaping from the organelle so that they may not be used in other biosynthetic pathways. These are some of the new approaches which have been incorporated into metabolic engineering to improve the efficiency of the metabolite production. Application of plant metabolic engineering. Let us examine some examples. One of the most complicated one is the incorporation of nitrogen fixing capability in the plants. That is, creating transgenic plants that can satisfy their need for fixed nitrogen by synthesizing their own nitrogen fertilizer, thereby eliminating the need for applying synthetic fertilizer. This is quite a complex process. About only bacteria are capable of fixing nitrogen. Nowadays, scientists are trying to incorporate this particular pathway into the plants so that plants can synthesize their own fertilizer, so that farmers are being freed from using synthetic fertilizers for crop production. There are 16 genes involved in this, and several scientists are using a minimum set of 10 genes to transfer to plants for production of nitrogenase enzyme. And this activity of this enzyme in particular biological conditions can fix atmospheric nitrogen into ammonium, which can be utilized by the plants as a fertilizer. In addition, recently a novel rice has been produced, which is known as golden rice. This is for enhancing nutrition quality of the rice. Bioengineering of beta-carotene pathway in rice produced high levels of precursor of vitamin A in edible parts and yielded the strain called golden rice. Vitamin A is very important for the vision, for maintaining in case of developing countries. Poor children, due to lack of vitamin A, they go blind at an alarming rate. So, scientists in the later part of 1990s developed a procedure to fortify rice with the vitamin A by incorporating the genes necessary for beta-carotene production. Initially, they used 4 different genes which are listed here: phytoin synthase, phytoin desaturases, and lycopene beta cyclases for conversion of geranyl diphosphate into beta-carotene a vitamin A precursor in rice. You can see in the picture here, there's a normal rice which is white in color, which is on the left side. And golden rice is yellow, that is a beta-carotene fortified rice. While enhancing nutrition quality, there's another important discovery is fortifying the fruits and the other edible parts of plant with the vitamin E, another very important vitamin for human health. For this, metabolic pathways in tomato as well as tobacco have been modified as an experimental basis to test whether vitamin E production can be enhanced or not. For this purpose, scientists have overexpressed homogentisate phytoltransferase and accompanied by two more enzymes, tocopherol cyclase and gamma-tocopherol methyltransferase. When these three enzymes are being overexpressed in tomato and tobacco, homogenous acid is converted into delta tocopherol and beta tocopherol, so on. And HPT, that is homogenous phytoltransferase, was found to be more active in fruits and in accumulation of tocopherols, whereas all three enzymes were found to be active in the leaves of tomato plant. This is one of the ways to fortify the fruits with vitamins. So we can utilize several different pathways to incorporate and production of different types of vitamins necessary for human health. One of the ambitious programs in recent past is increase the photosynthetic efficiency of plants for achieving higher productivity. In this, the scientists are trying to incorporate carbon dioxide concentrating mechanism from cyanobacteria, that is blue-green algae, and transfer the genes into plants so that carboxysome is produced, where in that compartment, carboxysome, carbonic anhydrase enzyme can concentrate the production of carbon dioxide and supply it to rubisco for enhanced carbon dioxide fixation, thereby eliminating the chances for photorespiration. This is a sort of optimizing the conditions for enhanced photosynthetic carbon dioxide fixation in the plants. This is also a very important aspect, and we expect that another 2 to 3 years such plant varieties will be produced with enhanced photosynthetic ability. There are several biopharmaceutical proteins which are being produced in the plants. For example, human somatotrophin, insulin-like growth factor, interferons, that's alpha interferon as well as gamma interferon, human proinsulin. Several different types of biopharmaceuticals are being produced in the plants and purified and supplied as components for use in health-related in the hospitals for improving the health of the humans. Another important aspect is engineering a biofuel feed that can self-destruct the lignin that interferes with the purification of fermentable saccharides. In this, what scientists are doing is lignin biosynthesis is genetically modified to reduce its content so that to improve the processing of polysaccharides. Lignin is the one which binds polysaccharides together, cellulose molecules, and when its production is being inhibited, cellulose becomes available for enzymatic hydrolysis and release of sugars, whereby sugars can be fermented for producing bioethanol and other biofuels. Metabolic engineering of plant secondary metabolites. What are secondary metabolites? Secondary metabolites are the organic compounds that are not directly involved in the normal growth, development, and reproduction of an organism, but are essential for their survival. Anthocyanins and flavonoids have antioxidant properties, and they are compounds of choice for higher levels of expression in food. For example, chalcone isomerase, an enzyme in the flavonoid pathway, was found to increase flavonoid production. When Petunia chalcone isomerase was overexpressed in tomato, 78-fold increase in flavonoid levels occurred in the fruit. There are several other examples, which include Arabidopsis, tobacco, rice, and maize plants, which lack isoflavone, which act as a phytoalexin in legumes to protect against microbes. Overexpression of isoflavone synthase in the plants which I have just mentioned, Arabidopsis, tobacco, rice, and maize plants, resulted in the production of isoflavone in the plants. Third compounds are terpenoids and alkaloids, comprising many drugs and poisons, are the largest group of secondary metabolites which are of significant value. While talking about bioengineering of secondary metabolites, some of the most important secondary metabolites are flavonoids and related anthocyanin production. Flavonoids and anthocyanin are antioxidants which are beneficial for human and animal health, thereby reducing the risk of developing cancer and other things. Recently, scientists have developed rice varieties where rice seeds have high amount of anthocyanin. Normally, in rice seeds, the rice we consume do not contain anthocyanin. Recently, there's a publication which says several genes have been transferred into rice and which confer the property of synthesis of anthocyanin in rice seeds, which is being shown on the right side of this picture, where the rice appears like pink in color. That is due to anthocyanin. I will not describe the details of this, but it's sufficient to understand that the rice and other cereals can be fortified with useful antioxidants like anthocyanin. Anthocyanin production has also been enhanced in case of tomato, as the picture shown here clearly indicates tomatoes which are purple in color. These are developed due to transfer of MIP transcription factors into them for the production of high amounts of anthocyanin, which are antioxidant substances and useful for human consumption. Besides flavonoids, there are other type of secondary metabolites, and metabolic engineering has been performed in the plants for the production of these type of secondary metabolites also, which are known as alkaloids. Alkaloids are a group of naturally occurring plant-produced organic chemical compounds containing basic nitrogen atoms in their core. They include many drugs like morphine, quinine, and poisons like atropine, stixine, which have physiological action in humans. There are two different types of alkaloids. Indole alkaloids, about 15 terpenoid indole alkaloids are industrially important, including anti-tumor vinblastine, vincristine, etc. The alkaloids share common pathway till intermediate strictosidine and then diverge depending on the plant species. Efforts have been made to overexpress the early genes to divert the metabolic flux into the alkaloid pathway. Strictosidine synthase, when overexpressed, results in higher levels of alkaloids. Alkaloid biosynthesis genes can also be expressed in non-alkaloid producing plants, such as cell cultures of Wiggly, where overexpression of STR and the tryptophan decarboxylase genes produced serpentine, a type of alkaloid. Isoquinoline alkaloid derivatives. This class comprises of pharmaceutically important medicines such as morphine and codeine. The enzyme scolarine methyltransferase is involved in biosynthesis of berberine. Overexpression of this gene in Coptis japonica resulted in increase in berberine production. This particular plant produces berberine as it is, but its overexpression enhances its production. When this enzyme SMT gene from C. japonica was overexpressed in cell cultures of Escherichia californica, which generally lack this gene, results in the production of columbamine. This is another type of alkaloid. Because of significant uses of this compound, efforts are being made for increasing the flux through biosynthetic pathways for overproduction of alkaloids. Another aspect of metabolic engineering is production of edible vaccines. Edible vaccines are bioengineered to contain the antigen but have no pathogenic effects. Transgenic plants express pathogenic genes which can elicit immune response but does not cause disease in humans and animals in the form of edible vaccines. These transgenic plants accumulate the foreign antigenic protein which can be consumed orally without purification from plants. This is done by transferring specific types of genes into plants and producing the proteins which act as an edible vaccine when consumed. First edible vaccine was produced in 1990, which has a surface protein from Streptococcus bacterium, which is done in tobacco. Second generation of edible vaccines are called multi-component vaccines, which provided protection against several pathogens, are also being generated. For example, second generation vaccines are based on epitope fusion of both subunits of chlorella, for example, cholera toxin. This cholera toxin epitope fusion protein also had a scaffold for presentation of epitopes of rotavirus and enterotoxigenic E. coli. This trivalent vaccine elicited humoral immune response as well as memory B cell and T helper cell responses. These are to confer the humans or animals ability to fight this toxin. There are different strategies to develop edible vaccines. Like any other strategy we described so far, a gene of interest need to be transferred to the plant and that need to be expressed, and a protein is produced which can be purified and used for either purified and used or directly consumed from the plants in case of edible vaccine. Either this can be done through transformation of callus cultures and production of protein and generation of plants, and the plant parts can be directly consumed, or plants can also, a gene of interest can also be expressed via viral vectors in the plants, and fusion proteins, epitope proteins can be expressed through viruses in the viral infection plants. These viruses are disarmed, they do not cause plant disease, whereas they produce proteins which are can be available vaccine. There are some examples of edible vaccines for disease are shown in this table. For example, vaccines against hepatitis B virus, Norwalk virus, rabies virus, and vaccine against gastroenteritis, hemorrhages, AIDS, and cholera. All different vaccines derived from the various proteins obtained from genes obtained from various viruses are expressed in several different types of plants which are listed here: tobacco, potato, lettuce, and so on, maize, to develop edible vaccines which can fight a particular disease or condition when they are consumed orally. Also, some therapeutic edible vaccines and diagnostic applications of these vaccines have been developed. For example, the vaccines which are used in cancer treatment, or like a colon cancer or infections to subvert herpes simplex virus, and so on. These are all again developed by transferring appropriate genes into plant system, such as tobacco, wheat, rice, and soybean, and also alfalfa. The diagnostic tools were also developed in the plants to detect the various types of complaints as diagnostic applications for these edible vaccines. So students, let us summarize this model. Today, I tried to familiarize you with the strategies of metabolic engineering to improve the production of high-value chemicals, therapeutic proteins, antigens, etc., using plants as production platforms. Other types of production platforms include bacteria and yeast, so on. Here our focus is on plants. In addition, we also discussed new trends in metabolic engineering research to incorporate enzymatic pathways to develop novel transgenic crops equipped with the nitrogen-fixing capabilities to synthesize their own nitrogen fertilizer, or to enhance plant performance by incorporating carbon dioxide concentrating mechanisms to boost photosynthetic efficiency and crop yield. These two examples I have already described to you in a little detail during my course of this lecture. Today's lecture presented to you with the latest trends in metabolic engineering hopefully will form a background or basis for you to improve upon this particular subject as per your interest. Thank you.